Okay, welcome to the third of these, the third of four sessions that provide an introduction to the elements of DEB theory with a particular focus on those parts that are relevant for ecology and ecotox. In last week's lecture, I worked fairly systematically through the basic components of that part of dynamic energy budget theory that dealt with the ability to relate the state of the environment through to patterns in growth and reproduction of a single organism. And in this lecture, I'm going to start by very briefly reviewing that material and then introducing a couple of other variables in the DEB theory that characterize the processes of aging and mortality, because we need aging and mortality as well as information on mortality as well as information on reproduction if we're going to do any population ecology. I'm then going to go very quickly through some concepts relating to the issues of estimating the parameters in the DEB model, but that is a sufficiently complicated matter that it goes beyond the scope of this particular course. I'm going to make a very brief comment about material fluxes, and there I'm deviating a little from the outline that I put out in the sense that this was going to be a major focus of this lecture. I decided I wanted to make a tighter connection to ecology because I'm going to need that in next week's lecture. And then much of the second part of this session will be devoted to some discussion of making the transition from individuals to populations and what I'm calling a DEB-based population dynamics. So let me briefly review what I did last day. The main purpose of the last lecture was to work through the components of Kooijman-Metz's standard DEB model. The basic idea is energy from food comes into a reserve compartment. Mobilized reserves are used for growth, reproduction, increasing maturity, and so on. And I broke the model down into various components, and I'm just going to put up 2 or 3 of the slides from last week's lecture as a memory prompt on what we did. We looked at the process of mobilization, what happened, the fate of mobilized energy, and the rule that determined how it was allocated between growth and somatic maintenance on the one hand, and maturity or reproduction and maturity maintenance on the other hand, and this was the so-called kappa rule. I talked quite a bit about the concept of reserve homeostasis because that was the key to understanding the mobilization flux. And a comment I got after that was that I might have mentioned a concept which you will encounter if you read chapter 2 of Kooijman-Metz's book, namely weak homeostasis, which is another way of getting at what I was talking about last time. And then I talked about the dynamic equations for maturity and reproduction. And again, this is the slide that I had last week. The main idea here was that the branch of the mobilization flux that was used for this purpose went to increasing the state of maturity in embryos and in juveniles, and in adults that energy was used for reproduction. And I ended the lecture with this fairly complex picture that illustrated how all the bits hung together, taken from the Souza et al. Paper in the Philosophical Transactions of the Royal Society, which I recommended as a good way of getting an overview of the dynamics of this system without getting overwhelmed with detail. There are two other variables that feature in any representation of the complete life cycle of an individual organism. I've highlighted 4 in the previous lecture, and I now want to say just a little bit about 2 further variables that appear leading to a set of 6 that in principle are the variables that we use to characterize a single organism. The slides up to this point illustrated the reserve, the reserve buffer, and hence the quantity we call reserve density, the amount of material in its structure, the amount of material assigned to maturity, perhaps it's not quite right to call it a buffer, and the reproduction buffer. If you read DEB papers, you'll encounter 2 other concepts. One is given the slightly mysterious name aging acceleration, and the second is given the name hazard rate, and that is a very familiar concept in ecology. It's just the per capita death rate, the probability per unit time of an individual dying. In many ecological contexts, a mortality rate is determined by factors unrelated to energetics, obvious examples being predation and disease. But what DEB theory does give us is a way of characterizing the physiologically related or the energetically related component of mortality, and it does so by taking advantage of the concept of damage-inducing compounds. The theory underlying this is described in Chapter 6 of DEB 3. That is one of the more challenging sections of DEB 3 to read, but the concepts are relatively straightforward. There's also quite a useful collection of little bits of additional information relating to this material in a file called Comments that Kooijman maintains at the website that is listed at the bottom of the slide here. That is actually, and this is a side comment, is a particularly useful place to a particularly useful file to be aware of. There are many places where I've had difficulty with some concept in DEB 3. I've gone to the concepts and I've found useful, very useful, additional information. It's an amazing piece of self-discipline that has had Bass maintain this set of comments, and in many ways it's for some purposes it's as useful as the book. The basic idea behind the DEB-related treatment of mortality is that metabolic processes lead to the production of reactive oxygen species, often abbreviated as ROS, and their immediate products. And the precise definition of what you count as a ROS is a matter of different conventions in different contexts. These in turn lead to damaged macromolecules, damaged membranes, and these in turn lead to increased risk of mortality. And the figure on the left that illustrates one way in which this can be thought of is lifted straight from the comments from the comments file that I've listed at the bottom. And as with all the other concepts in DEB, we require a level of abstraction. Just as we reject talking about lipids, proteins, and so on, but talk about reserved structure, likewise, instead of directly talking about a ROS and derivatives, we adopt an abstract representation where we think of 2 types of compound. One is called damage-inducing compounds, the other is called damage. And in next week's lecture, which is on ecotoxicological applications, I'll talk a bit about how you relate the damage-inducing compounds to real measurements. I've introduced a state variable which I've called uppercase Q, which is the total mass of damage-inducing compounds in an organism. And just as we defined reserve density, we can define damage-inducing compound density by simply dividing Q by the structural mass. and this quantity, lowercase m with a subscript q, is the variable that is used in chapter 6 of DEB 3. Likewise, we can define a quantity big D, which represents cumulative damage, and a density little m sub d, which again is the variable that is used in DEB 3 as being that fraction per unit volume, per unit of structural mass. I've deliberately written it down in terms of these quantities that don't take account of density, because then it's rather easy to identify the key assumptions that go into deriving the equation for the density. The idea is that there are two potential contributions to the rate of production of damage-inducing compounds. The first is directly proportional to the mobilization flux. That is, the faster metabolic processes lumped together are going on, the faster, the greater the rate of accumulation of damage-inducing compounds. That's really just saying that an organism that is doing lots of metabolic things fast is producing more more metabolic products. And the second term recognizes that there can be feedback between the presence of damage-inducing compounds in an organism and their production. And in most cases considered so far in DEB applications, this has been positive feedback. That's to say, the more damage-inducing compounds there are, the more are produced. So there is, in a sense, an acceleration of damage. But in at least one example in DEB 3, Kooijman finds a negative exponent in there, which is tentatively interpreted as a repair mechanism. Once you have that equation for the rate of change of Q, you can very easily move to an equation for the rate of change of little m, the density of damage-inducing compounds, and the only difference between the equation I've written down in appearance and the one that then appears for little m is that there is a dilution due to growth term that appears in the equation. The next assumption is relatively straightforward, that there is some rate at which damage-inducing compounds are turned into damage, and the hazard rate, that's the per capita risk of mortality, is assumed proportional to the damage to the damage density. And what we see here is that the end result of this is that we are getting a link between mortality rate, the hazard rate, through this quantity MD. That in turn is linked to the mobilization through this term J sub EC in the earlier equations, and which is the mobilization flux. Consequently, there is a connection to mortality. It's a delayed effect. Increased mobilization does not lead directly to a change in mortality. Increased mobilization leads to a change in damage-inducing compounds that in turn leads to a change in mortality. As always, when an abstract concept is developed, I've been demonstrating how it applies to Daphnia, and in lecture 1, I showed you this picture, for which the axes are probably not too clear on the screen. The x-axis represents let me get myself a pointer, the x-axis here represents age in days, and the y-axis is the per capita mortality rate of an organism, that is the hazard rate. And the end result is that the higher the food density, the higher the per capita mortality rate. That's to say, organisms experiencing low food but still sufficient for subsistence were capable of living till close to 100 days, while a very few achieving the highest food lived beyond 60 days. And that picture, circled in red in the next slide, is actually is completely consistent with the formalism that I've just described. The higher food animals have faster metabolism, hence increased damage-inducing compounds, increased damage faster. The weakness in this is that there is a region down here that I'm highlighting, circled in red in the curve, which represented mortality in the early life stages of the animals, where the trend was in the opposite direction. That is, the poorly fed animals were the ones that experienced the highest mortality. I've highlighted it here because you'll see in the end of the second part of this session that what appears to be a few data points down there turn out to be rather important for understanding what's going on in population dynamics. So to pull this together, we have theory that links metabolic rate through to mortality, in the sense that it describes an increase in mortality associated with faster metabolism. We don't, up to this point, have theory that characterizes, in a sense, the behavior of starving animals. And there is a reasonable argument that says, to some extent, this type of behavior may well be organism-specific, that there is potentially less generality there than in the other areas, but that is than the other mechanism. But that's a matter that is open for discussion. So now we have completed with that little overview of the mortality processes. I've completed my characterization of the 6 variables that are involved in the DEB representation of a single organism. I've completely ducked the question of how we get at the parameters in these models, and in many ways, this is the key to applying the models. And there are 2 seminal papers, I think, in terms of understanding how this is done, and it is again a theme that goes way beyond the scope of a set of introductory lectures. There is a paper highlighted here by Bas Kooijman and about 4 or 5 other co-authors that came out 5 years ago. And this paper focuses at the level of one species, and it asks the question, what information do you need in order to get at each of the many DEB model parameters that we've looked at? What can you learn from minimal information like a maximum size and von Bertalanffy growth rate? What more can you learn if you have a set of growth curves at different food levels? What more can you learn if you have some information about chemical composition, and so on? It systematically runs through a number of steps telling you what extra information you can get from different types of additional information. It offers a systematic recipe for relating model parameters through to the sorts of data that are commonly available. However, a major advance in parameter estimation came with theory that was published in a paper that came out in 2011 that took advantage of Another distinctive feature of DEB theory that I have mentioned at about 4 or 5 points in these earlier in these lectures, but have not emphasized, and that is that the model parameters, it is possible to predict how the model parameters vary interspecifically, that's among species, and I'll elaborate just in a minute about how that works. But because DEB theory provides a foundation for an explanation of what are sometimes called body size scaling relations, how various rates vary with size, like the von Bartholantzi growth rate or the respiration rate that I showed last time. Because DEB theory provides that foundation, it's possible in principle to use information from other species in order to estimate the parameter values for a particular for a particular species. In particular, suppose we have a collection of moderately functionally similar organisms, like for instance, a collection of mammals of different size. It's well known that there are empirically observed relationships between different metabolic parameters for these. DEB provides an explanation in terms of the model parameters, and that in turn means that we should be able to use information on the physiological properties of a mouse to estimate the DEB properties of a rat, and so on. And this paper by Lika et al. Expands that. I'm going to elaborate on that in just a second, but then the final point I would make is that largely because of the valuable work in the Lika et al. Paper, there is increasing appreciation of the value of Bayesian approaches for estimating model parameters, where information on other species provides you with some prior estimates of your parameters, and then you use the information you have to refine it. That is very much for the future, but I've mentioned one paper with Leah Johnson as lead author that demonstrates the potential for Bayesian methods of DEB parameter estimation. I want to make a few comments about the challenge of estimating parameters, and in many ways the starting point is recognizing that there really are two types of DEB parameter. There are the parameters that appear in the models, and these parameters relate to the relationships between quantities like reserve, structure, damage-inducing compounds, and so on. And all of these are rather abstractly defined. And then for any particular organism, there is of necessity there are some parameters that relate these abstract quantities to things you can measure in the real world. If we don't specify, except as a generalized compound, what reserves and structure are, we need some rule, which means some parameters that relate the amount of reserves and the amount of structure to, for instance, the mass or the total carbon or the total nitrogen or whatever. And these are what we call auxiliary parameters. And to illustrate that the interconnection can get really messy, I've just pasted onto the bottom of this slide a picture from the Lika et al. Paper that illustrates just the complexity of the complexity of the process. I think it is greatly helped by keeping separate in your mind these two concepts, the core parameters and the auxiliary parameters, because if you want to stay within the framework, you have a lot of species-specific flexibility with the auxiliary parameters that you don't have with the core parameters. So let me outline briefly how the covariation method, which I think could be thought of as a pseudo-Bayesian method, works. Like I said, it exploits the DEB model predictions on the interspecific variation of parameters. And I like to think of it as doing so in terms of some jargon that was very common in the DEB literature a decade ago, seems to have gone out of fashion, but I find useful, and that is to distinguish what are often called intensive parameters and extensive parameters. And the terminology is loosely analogous to the use of these terms in thermodynamics. Intensive parameters are parameters that have broadly constant values across species, and these are parameters like after temperature correction. So these are parameters like the specific maintenance rate, the energy required per unit time to maintain 1 unit of structure, the energy conductance, which relates to transit through the reserves. These are assumed to be based on fundamental physiology that is broadly similar across many species, and they have roughly the same value. There's a second set of DEB parameters that roughly scale with the maximum length or some other measure of size of the organism, and an example of these would be the parameter in the specific in the specific assimilation rate. So the rate at which the parameter that relates the rate at which a whale feeds as a function of length to the parameter does not have the same value as the parameter that describes how the feeding rate of a Daphnia varies with its length. But there is a systematic size dependence of these parameters related to the difference in maximum size of a Daphnia versus a whale. And this theory takes advantage of these. And what that means is that if we have obtained parameter values for a bunch of species, and let me stick with my example, a set of mammals, because I'll show data from mammals in just a minute. If I've collected these, and you come along with data for a new mammal for which nobody has estimated the DEB parameters, we can have a first cut at this by simply imagining that the new mammal is a scaled-up version of the average of all the other mammals we've seen. And this gives us a starting point. And then the idea in the covariation method, as developed by Dina Lika and collaborators, is that you now look for your estimate of the parameter as a weighted sum, for instance, of the of the deviation of the parameter values from essentially a residual sum of squares calculation, but one in which you pay a penalty if your parameter values differ a lot from what you would expect based on the information from other species. And that in turn lays the foundation, I think, for future work where which would use true Bayesian methodology, where you would be constructing a set of prior distributions and working from that. One recent piece of work that convinced me that this approach really has a lot of power is a piece of work done by James Mino, relatively recently, and the paper on this is currently in review. And what James did was looked at data for a collection of mammals from a large data repository called AdMyPet, which Bas Kooijman keeps and can be accessed through his website. And those who are doing the telecourse that starts today, in fact, will, as a major exercise, will be required to add their pet to the collection of species that are out there. But James Mino used these data to estimate to estimate the metabolic rate of how the metabolic rate of an animal of a mammal would vary with its size. So these were parameters that were estimated from information like a maximum size, offspring production, embryo properties, all the bits and pieces that go into estimating DEB parameters. And then he looked at the mean of these parameters, looked at the formula for respiration, and actually predicted what the respiration rate would look like. And the red curve here that I am highlighting with my pointer now was what was predicted, and the data points that you see are data from some of the best known data sets out there, which are the variation of respiration rate with size. The broken lines represent confidence intervals based on the variability of the estimate of the individual DEB parameters that he came up with. And it's obviously not quite spot on. In particular, there is a bit of a problem at the lowest end, but it tells you that this information based on covariation of parameters actually gives you potential for making some upfront predictions rather than simply retrofitting rather than simply retrofitting metabolic data. So up to this point, I have completed the description of the DEB model by adding mortality. I've given you a few pointers to the challenges in parameter estimation, and this will be a major focus for anyone who continues and works on Bass's telecourse. The remainder of this opening session and the entire second part of this lecture is going to be devoted to talking about how we make the transition from this information through to population dynamics, and that will be mainly in the second part. But I want to use the remaining 6 or 7 minutes of this lecture to set up some preliminaries targeted primarily at anyone listening to these lectures who's not primarily an ecologist and hasn't thought much about these questions. And what I want to do is, by way of preparation, is present just a little bit of demography. This is what you would see in an Ecology 101 type class. Suppose we have an animal population. Let's suppose the sex ratio is constant and we focus on the females. Let's imagine that we divide this population into a set of discrete age classes. So if we're dealing with a long-lived organism, it could be all the individuals aged 0 to 1, 1 to 2, 2 to 3, and so on. If I use the symbol S with a subscript A to represent the fraction of newborns that survive to age A, and I use the symbol beta with a subscript A to represent the total number of offspring that survive to age A, then I can define a quantity which is often given the symbol R0, and in fact is often referred to by ecologists just as R0. R0 has become a name that is used. And this is the average number or the expected number of female offspring expected in a lifetime from an individual, from a newborn individual. So you imagine a newborn individual. It has a probability SA of surviving to age A. If she survives to age A, she will produce beta A female offspring. So now you just take a weighted sum of all of these and you get the total expected number of offspring to be produced in a lifetime. Somewhat misleadingly, that a quantity is often called the net reproductive rate in ecology textbooks. I find this frustrating since it's a number and not a rate. If anyone's interested, they will also discover that this quantity is very frequently used in epidemiology. In that case, it characterizes the number of new infections that are produced by a newly infected individual, and it gives you a measure of the virulence of a disease. DEB theory works in, I've set this out as a summation because this is the form in which it appears in many, many ecology books. Sometimes the symbol L is used rather than S for survival and the symbol M rather than beta for offspring production, but this summation formula is the way that it's presented in most demography textbooks. DEB theory works in continuous time. We have used differential equations. All that happens is that with a slight redefinition of beta, we get an integral instead of a sum. So the quantity R0 is just the total. It's just the total. It's still the average number of offspring produced in a lifetime. And the link to standard DEB occurs by recognizing that the equations that we have developed, all 6 of them, actually give us a recipe for calculating the quantities S and beta for any organism. And they're messy differential equations, but they're pretty easy to code up in Mathematica MATLAB or whatever your favorite software package is. And going back to the start of this lecture, the first 4 state variables that I described let you calculate the reproductive output as a function of age. So these 4 tell you the quantity, how beta varies with age. The 2 that I didn't write down explicitly for the aging acceleration, which is related to damage-inducing compounds, and the hazard rate that is related to damage, these 2 equations let you figure out the survival to an individual aged A. So now, if we solve these differential equations, we have a reproductive output as a function of age, we have a survival as a function of age. We can now just perform the integral again numerically, and we get to R0. So the theory that we have developed up to this point lets us calculate at least one of the fundamental quantities in basic ecology, which is the net reproductive rate of a particular organism in a particular environment. Of course, population ecology is interesting simply because most organisms do not grow, and most populations do not grow and decline exponentially. There are various feedbacks and interactions that lead to population regulation, population cycles, and so on. And I'm going to demonstrate in the second portion of these lectures, after our 10-minute break, how that comes together and how we can use the DEB-based formalism to think about a realistic population dynamics. So a 10-minute break, and in my time we will resume again at 9:50. Thank you. Thank you. I'm going to restart. I apologize for that. I duly started talking away without turning on my microphone, and I gather it's now working. So what I did just before the break was to review some basic demography, which led us to the understanding that if we simply consider fixed demographic rates, an unchanging environment, the ultimate fate of our population is either exponential growth or extinction. Again, if there is no feedback, I didn't show this, but it's well known that this result that ultimately you get exponential growth or extinction persists even if we take account of environmental fluctuations. And in continuous time, rather than the quantity R0 that I talked about, it is convenient to work with a quantity commonly represented by little r in ecology textbooks, which is the solution of the Euler-Lotka equation that I've written down here. It's not an equation that you can generally solve analytically, but again, it has nice mathematical properties. It is very, very easy to solve numerically. And note that it is very similar to the equation for R0. The only difference is this exponential term in here. What changes things and makes everything more interesting is when we have feedback. If we have feedback with the property that when the population is larger, either the survival goes up or the fecundity goes down, then we have the potential of getting some kind of equilibrium population, and in an equilibrium population, the quantity big R naught is equal to 1, or equivalently the quantity little r is equal to 0. I mentioned before the break that we could compute the quantities beta and S by solving the DEB model equations. If you want to do this and actually get the long-run growth rates or study equilibrium populations, there is some very convenient software in a paper published 4 years ago by André de Roos in Ecology Letters. He describes a computational approach, and he includes in the appendix material a software that does this for a simplified version of Bass's DEB model. It would require adaptation to work with the full model, but all the hard stuff, I think, is done there in this André de Roos paper. Let me illustrate these concepts with just yet again example from Daphnia, and the top curves represent beta A, the lower curves represent survival, and we see that at different food levels, at the highest food levels you get the highest fecundity and the shortest lifetime. And I've put that up to illustrate an important property, which is that both the quantities R0 and the quantity little r, which after all are really just two ways of expressing the same property, vary very strongly with food availability. The Daphnia in the picture subsist on microalgae. The x-axis is the algal concentration on a log scale, and what you see is a very strong dependence of a growth rate on food concentration. And a point I want to highlight is that if you're interested in ecological applications of the model, the food densities that we're going to be concerned with are often way down at the low end of the range for which lab information is available. And this is yet another reason why the model is, why having the models are important, because there's far more frequently data in this range than in this range, and this is what we need if we want to understand natural populations. This is just an indicator of the range of edible algal concentrations that are commonly observed in, say, the clear water phase in a temperate lake. If you want to relate the quantities we've derived to data, one immediate question to ecologically relevant data, one immediate question is whether you really need to study the animal for the whole lifetime. And the answer broadly is that the faster the population growth rate, the shorter the length of time over which you need data. That's to say, if you study the growth of Daphnia, in that case at a high food level, and you plot out the estimate you would get for little r by terminating the integral at 21 days or whatever, let's take 21 days here. For the highest food level, you would get pretty much the correct answer. At the lowest food level, reproduction wouldn't even have started. And I mentioned this because in next week's lecture, I'm going to illustrate the application of DEB to interpretation of ecotoxicological data, for which much of the information comes from 21-day chronic toxicity tests, and there is a series of issues. So how then do we incorporate the feedback from the living organism to the environment? There's essentially two ways of doing this. One is to use what are called structured population models that I will describe in a minute, a family of mathematical models that are widely used in ecology. But the most intuitively obvious way of doing it is to recognize that a population is nothing more than a collection of individual organisms that are interacting with a shared environment. If we want to construct a population model based on dynamic energy budget theory, we imagine that the individuals are all the individual organisms are following the rule of the DEB model. So, in principle, all we need to do is throw a lot of individuals into a computer, let them interact with the environment. Every time they produce an offspring, we create another variable, and that interacts with the environment, and so on. And that way we can follow a population dynamics. And the remainder of this lecture is going to be devoted to saying just a little bit about how each of these two approaches is being applied in a DEB context. The great advantage of the approach based on structured population models is that there is a vast body of mathematical theory that has been developed relating to structured population models. There's therefore a lot of dynamical understanding that we can build on. The great advantage of individual-based models is their conceptual simplicity, and the fact that we can work in a more detailed way and add system-specific features as needed. One comment about the structured population models, again a reference to André de Roos on his website at the URL link there. You can get hold of some very powerful software called the Escalator Boxcar Train that again lets you perform numerical computations with structured population models. A detailed remark, if anyone followed the lectures I gave 2.5 years ago on this subject and is following and still has the slide from that, the link to the EBT has changed over the past 2.5 years, and what is down there is the current link. So let me say a little more about structured population models. As I've said, they emphasize dynamics. They're mathematically sophisticated, and but they let us describe population growth, regulation, invasions, life history, evolution, species. In fact, all the topics of an introductory ecology class. Just like DEB, they are abstract, and they involve a sophisticated treatment of what mathematicians call the P-state, which is the function that you use to characterize the population, which could be its age distribution, its size distribution, a joint size and age distribution, and so on. But for our purposes, I want to emphasize that there are two important situations. One is a situation where we can approximate the population dynamics as being essentially zero-dimensional. That means it's just a set of variables that represent population size or mean value, some quantity or whatever. And then I want to point to one particular example for which a lot of work has been published very, very recently that makes really a very nice connection to what we've been doing, which is the Kooijman-Metz model. Let me elaborate on how you get ordinary differential equations to describe a structured population model. That title is deliberate because you will often read the misleading statement that if you describe a population by a set of ordinary differential equations, you are assuming that all individuals are identical. You are not. In a DEB context, you're assuming that they all have physiological rates that are proportional to biomass. There is language in the DEB theory book that calls an organism of this sort a V1 morph. So if you have exclusively V1 morphs, and repeating organisms whose physiological rates, that's uptake rate, respiration rate, well, uptake rate, maintenance rate, and so on, that is proportional to structural volume, you can commonly derive a simple set of ordinary differential equations. The other potentially very restrictive assumption, but not as bad as it might appear, that you have to make is that all organisms experience the same hazard irrespective of their state. So a small organism versus a large organism of the same species, a young individual, an old individual have to experience the same hazard rates. So if you make that assumption, you can actually derive from the DEB equations a set of ordinary differential equations. The assumptions, I'd say, are defensible for a few situations. The largest number that I've seen published relate to microorganisms in a chemostat, and I'm going to show you a couple of examples, or a few pieces of data that relate to an animal population, which again is, of course, Daphnia. But again, these simple models are just that little bit more more valuable than they might appear, and the reason is that they're structured. They're, albeit in a very simple way, capturing quite a bit of the DEB complexity without incorporating all the surface to volume features that, of course, are an interesting part of DEB. The next two slides are lifted from the Ecological Dynamics, a textbook, and they're there. The details don't matter. They're there simply to demonstrate that simple population models using ordinary differential equations are consistent with some population data. These are situations where an equilibrium population was predicted with one of these, and these are field data where the density of edible algae, that is Daphnia's food, in lakes was predicted using one of these simple ordinary differential equation type models. And there's a lot of scatter and so on, but it's getting into the right ballpark. Especially interesting from the perspective of DEB, however, is that you can go beyond simple biomass models. You can bring reserves into the ordinary differential equation representation. And the data in the figures here are data from a 1970s paper I'm having a senior moment and forgetting the name of the name of the author of the original paper, but these were data on a chemostat system with a glucose substrate, a bacteria that were energy limited, so supported by the glucose, and a predator, which was a slime mold, which was subsisting on the prey. The model was fit to an ordinary differential equation model based on the standard DEB model, but assuming that all individuals were a V1 morphs, meaning everything scaled with a structure of volume, and that mortality rates were independent of age, and that's particularly reasonable in a chemostat where the primary source of loss of individuals from the system was actually washout. And two curves are fitted to the data. The curves that are the bad match to the data actually just model biomasses in this system. They follow the simplistic approach that did rather well with the Daphnia. They actually get the phase relationships badly wrong. The continuous, or the bolder lines that I'm showing here, here, and here, and so on, and likewise over here, are fits to a model that took account of the reserve. So the addition of the reserve variable was the key in this particular case to getting a good match of a model and data. And with that feature in, but many other features missing, and hence getting to the ODE model, these data were pretty well fitted by a DEB-based model. I think it's worth emphasizing, though, that there is a structured population model for which there is now a large amount of information available that gives us a really nice intermediate situation between the full DEB model and making the assumption of ordinary differential equations, and that is the so-called Kooijman-Metz model. It's based on a model that actually predates DEB, that was published in 1984 by Bas Kooijman and Hans Metz. We can think of it as being just a model where an individual organism is characterized by the standard DEB representation, except we're short-circuited out the reserves. We don't even think about the reserve at this point, and we have removed the concept of maturity. So this slide just contains my much repeated picture of a standard model of growth and reproduction, but with certain portions grayed out. If you do that, what has happened to my slide? Yes, if you do that, you get a set of equations which I have copied here from a book that has very recently come out by Andrew DeRose and Leonard Persson on what they call population and community ecology of ontogenetic development. Essentially, it's a study of population and community ecology based on very much the same ideas that we have been developing in this course, but focusing on size structure rather than more complicated physiological structure, but still working with biomass-based models. In that book, they use extensively some very simple models, which they then compare with the Kooijman-Metz model. And the Kooijman-Metz model has a series of dynamic equations which I don't want to describe in detail, except to note that they follow very much the standard formalism of structured population models, which you can read about elsewhere. And then they have a series of assumptions which actually have a lot of overlap with standard DEB. They have a function that describes resource ingestion rate. In their notation, length is lowercase l, so ingestion rate goes as the square of length and multiplied by a type 2 functional response, because little r represents the density of resource here. They have an equation for the consumer growth in length, and that is just the von Bertalanffy form that we have been looking at again. They have an equation for consumer fecundity, which is actually the equation, if you go back to my first lecture, that we got through thinking about what I call the assimilation allocation, and that in turn is the same as you will get from the Kooijman-DEB model if you neglect reserves and neglect maturity maintenance. Then there is an assumption about a background death rate which is not energy related in at least the simple version of these models. And then there is a weight-length relationship which assumes that the total weight of an organism or total mass of an organism is proportional to the cube of length, which is again intraspecifically consistent with DEB. So the 4 red arrows highlight the features of DEB that this model is characterizing. I would not have emphasized it in an introductory lecture course, but for the fact that this book has now appeared, and this book contains a considerable number, probably a couple of dozen examples of ecological phenomena, ecological properties that have been worked out using the Kooijman-Metz model. So if we are faced with any population problem now where we want to get some intuition about what might go on before getting into the complexities of DEB, there is a huge amount of information available, and much of it is summarized in this fantastic book. The second approach to modeling a DEB-based approach to modeling populations is the brute force approach of using an individual-based model, and there's been a major advance in this area over the past 2 or 3 years due to the work of Ben Martin, and I want to thank Ben for teaching me everything I know about this model, and even for lending me some slides that were adapted for this remaining part of the course. And Ben's work essentially pulls together the insight in 2 books. One is DEB3 that we've talked about many times. The other is information from a book by Volker Grimm and Steve Railsback. A book title is here, and Volker is in fact Ben's PhD advisor, and Ben's work involves pulling these different insights together. In brief, the work in this book provides a framework for individual-based modeling. The DEB theory provides the representation of individuals for the work. And this slide illustrates essentially how DEB-IBM works. Of course, it's illustrated with regard to Daphnia, but here we have an adult female Daphnia. The rules of the model describe how she produces a bunch of smaller Daphnia. These in turn either die or mature to become more female Daphnia, and you simply keep track of the growth and reproduction of every animal in a population, simulating, say, several hundred of them, by brute force computation. Nice features of DEB-IBM are that it's implemented in a relatively easy-to-use computing environment called NetLogo. It lets us, when we're working with simple environments, and by simple I really mean non-spatial, it's particularly easy to do the NetLogo programming. And that is in large part because of a paper for which Ben is the lead author that gives a full description of the implementation of a DEB model within the NetLogo environment, and there is code that can be downloaded for that. So it really is a very powerful tool. I've used it for one or two problems in a context where I had no personal time available to spend programming and just time to tweak things. And as says here, there is a user manual that helps you get going. If you run DEB-IBM, you will finish up getting a screen display, something like this. And again, the details don't matter, but what you're seeing and what are illustrated in the top here are the sort of output variables you can keep immediate track of while you're going. In this case, these are the populations of three different size groups of Daphnia. In the middle curve here, it's tracking the food density. In the bottom curve, it's just calculating what is often used in ecological, what you often see in ecological paper, which is just the total population density, adding up the number of individuals. But you can plot anything you like. Especially interesting and useful is the fact that it lets you keep track of variability among individuals within the population of various characteristics. And these are just histograms at some snapshot in time of different properties across the population. You change parameters by working in these windows, and it's relatively straightforward to change what you want to be in Windows and not. That just involves a little bit of NetLogo coding and so on. Ben did some work in which he parameterized a DEB model for a individual Daphnia using data on individual growth and reproduction from the lab of Thomas Preuss in Aachen, and then took the standard DEB model and tried to predict what a population would do. And the Preuss lab also had data on a population dynamics in what I call a semi-chemostat. That's like a chemostat in which there is constant input of nutrients, outflow of nutrients and microalgae, but the system is closed as far as the Daphnia is concerned. And the data, these were replicated experiments, data from the mean and extremes of some experiments are shown here. The model is shown here and here. These are abundances of this is total number of individuals, this is the small individuals, this is medium individuals, this is large individuals, and the story is that the model does horribly. If you simply extrapolate from the mean population, if you simply extrapolate from the data on individuals, take the standard model and try to predict what happens to a population, the news is bad. But now you can do what is, if you like, standard practice in ecological modeling. You can look at the model and try and figure out what is missing. And it turns out that the primary problem here is one I highlighted some time back, which is that starvation and the response of animals to low food, and in particular juveniles to low food, is not well characterized by the model. So the next step in the work was to try to characterize and parameterize the response to low food. And when that was done and added into the model, the data and model come together like that. And what has then happened is that we have, in this, taken a model, an off-the-shelf, if you like, the off-the-shelf DEB model, parameterized it, applied it to a population problem, used the mismatch, and mismatches of model and data are often at least as instructive as matches of model and data, used the mismatch to identify a key missing ingredient, and when that was done, brought data and model into remarkable congruence. And that would be all very fine if it was the whole story, but a really nice part of the story that for me illustrates the fact that you can take a DEB model, apply this kind of fine-tuning, and then reach something that you would not otherwise have reached, is that the model offers insight into completely different population data from the ones that were used in anything that I've mentioned up till now. Ed Macaulay, a longtime collaborator of mine, ran a set of experiments in both small aquaria and large stock tanks over many years of interacting Daphnia algal systems. And the two slides at the bottom, on the left-hand side of this figure illustrate two common patterns that were observed. One was that the Daphnia population cycled, and they cycled with large amplitude cycles and cycles whose amplitude, if anything, grew over time. So the population was exhibiting very much standard boom and bust population cycles, such as is described in a lot of population ecology textbooks. The alternate possibility was that the cycles damped out, and you were left with approach not quite to an equilibrium, but to a pattern of small amplitude cycles. And that's just two pictures, but I would mention that there are many other examples of these two types of patterns, including some described in an earlier paper from the Macaulay lab that talks about transitions from one type to the other. These cycles are characterized by a property that we can get at directly using DEB theory. If you look at the cycle period and then look at the rate of growth and hence the time to reproductive maturity of the individuals executing the large amplitude cycles, what you find with data is that the populations in the large amplitude cycles for most of the time grow very rapidly and reach reproductive maturity very rapidly. By contrast, the populations executing the small amplitude cycles have really slow juvenile growth and reach reproductive maturity after a long time, and the cycle period is somewhere in between. This phenomenon has been described in terms of more traditionally formulated population model, state-structured population models based on delay differential equations. In fact, there is one such model in this paper here. But what Ben Martin did now was illustrated by some pictures here and here representing two portions of a long population trajectory. He now ran the DEB model, or the DEB IBM model, and depending on random stochastic factors, he either got the small amplitude cycles or the large amplitude cycles, and sometimes got a transition from one to the other. And now, during the period where the cycle amplitude was increasing, during the transition to the small amplitude cycle, he plotted both the mean and the extremes of the maturation time, that's the time to reproductive maturity as described by the maturity variable in DEB, and then followed it through the other way, and as he went to the small amplitude cycle, he saw it increase above this line, which is the average cycle period over the run. In other words, the DEB model, parameterized and modified based on Price's data, was capturing the complex demography of Ed Macaulay's experiments. It was also demonstrating the robustness, as it were, of the ecological mechanism, because there was another model based on size structure that also captured this demography. But essentially, for the purposes of this lecture, we could take the DEB framework, go through this systematic process of model formulation, modification, testing, and then moving to a brand new context and getting some getting some enhanced explanation of existing data. So I think where this leaves us is that we now actually have, in a way that what I couldn't have said a couple of years ago when giving a set of DEB lectures, we actually now have some really powerful tools for both the structured population modeling approach and for the DEB IBM approach. We have Ben Martin software for the DEB IBM. We have the large body of insight from the DeRose work on the Kooijman-Metz model, and together, I don't think it's a business of choosing one versus the other. I think it will often be most instructive to work with both and move back and forward. But we actually have the potential for making very rapid progress, I think, now in a range of population contexts. So stepping back, to wind this up, the main messages from this second lecture are that where there is no feedback from organisms to the environment, a population grows or declines exponentially. That's basic ecology. The rate of exponential growth can be calculated using DEB theory from information on the complete life cycle. When there is feedback, we require an individual-based or a structured population model, but we're now in the situation where there's good software available for both, and as I've said, a good choice of structured model. With special assumptions, structured DEB-based models reduce to a system of ordinary differential equations that include the potential for taking account of the reserve variable, and you saw the microbial example where that worked. And in some situations, DEB IBM results are consistent with previous work on size structure, and that potentially gives support for both ways of thinking. And that's as far as I want to go on population dynamics. Week 4 of this session will be devoted to applications of the theory that we've been talking about to problems in ecotoxicology and nanotoxicology, with particular focus on some of the problems involving environmental implications of nanomaterials that are the subject of people working in the CEIN. Thank you.